On October 7th, the Nobel Prize in Medicine for 2024 was awarded to two American scientists, Victor Ambros and Gary Ruvkun, for their discovery of microRNA. In this video, we will try to understand what exactly microRNA is, how it works, and how the discovery of microRNA has revolutionized our understanding of gene regulation. We'll also answer how a single cell zygote develops into a multicellular human, and in the end, we'll try to answer how it can potentially cure cancer. Before we get into microRNA, let's zoom out for a second. Think about your body. It is a complex system made up of different organs. These organs are built from trillions of cells. Now inside each cell, there are various cell organelles, and one of it is nucleus. And as we say, mitochondria is the powerhouse of the cell. Similarly, we can say that nucleus is the command center of the cell. Inside the nucleus, we have chromosomes, 23 pairs of chromosomes to be accurate. These chromosomes act as a package that holds your DNA, which contains your genes. These are blueprints for everything your body needs to function. Every cell in your body, whether it is muscle cell or a nerve cell, carries the same DNA. Yet, each type of the cell performs different tasks, and this is where gene regulation comes in. It ensures only necessary genes are switched on in each type of cell. In the past, our understanding was that this regulation mainly happens during transcription. During transcription, the DNA inside your gene is copied into a molecule called messenger RNA or mRNA. Now remember, this is different from microRNA. These mRNAs carries the instructions out of the nucleus like a courier delivering blueprints to the factory floor. In this case, the factory is ribosomes, where proteins are made. Ribosomes read the mRNA and assemble the proteins based on the instructions. These proteins are crucial for everything your body does, from repairing cells to sending signals between neurons. However, not all instructions are needed all the time, and producing too much or too little of certain protein can disrupt the delicate balance of your body's functions. So there was a gap in our understanding. This is where microRNA comes into play. Back in the early 1990s, Ambros and Ruvkun were studying a tiny worm. Now the name of the tiny worm is little difficult. Habitatis elegans. Most probably I have pronounced it wrong. Anyways, despite its small size, the worm was a perfect model organism because of its simple structure, transparent body, and a short lifespan that made it an ideal candidate for studying genetics and development. During the research, Ambros and Ruvkun made a shocking discovery. They found that a tiny molecule which was just 22 nucleotides long was acting as a key regulator in worms' development. This molecule wasn't coding for a protein, as many RNAs do. Instead, it was binding to the messenger RNA and preventing it from being translated into protein. This tiny regulator was microRNA. MicroRNAs are small molecules that act like regulators or fine-tuners in this process. They are created in cell's nucleus and processed by enzymes into their active form. These small molecules are like quality controllers. They bind to mRNA and either block it from making too much protein or stop it entirely. This process helps control the amount of protein produced in the cell and ensures that proteins are made at the right time and in the right amount. Without this critical control, things go wrong, leading to diseases and developmental issues. Without microRNAs, your body will fall into chaos. And now imagine if muscle cells suddenly starts producing nerve proteins. That would be funny, but catastrophic. MicroRNAs prevent these types of mistakes from happening, making sure that each cell sticks to its particular rule. Now this is a very simple explanation. Actually, this thing is a little complex, but I hope you have understood what microRNA is and how it works. Coming back to the research, Ambros and Ruvkun's discovery didn't just add a new layer of complexity to gene regulation. It changed the way we think about the development entirely. For decades, scientists believed that the gene regulation was mainly about turning genes on and off at transcription level. But now we know that there is much more going on behind the scenes. Post-transcriptional regulation through microRNA allows the fine-tuning of gene expression. After the initial copy of RNA is made. This fine tuning, as we have learned, is essential for cell diversity, development, and function. Now let's answer the question: how does one-celled zygote develops into a multicellular human? All the cells in your body have exact same genetic information. Yet some cells become muscle cells, others become nerve cells, and some become skin cells. But how does this happen? The answer lies in microRNA. These molecules help ensure a right gene expression depending on the type of the cell and its function. This is called cell differentiation, the process by which single fertilized egg develops into a entire organism. Now let's talk about the other side of gene regulation. While microRNAs are essential for healthy development, when their regulation goes off track, it can lead to diseases, particularly cancer. Scientists have discovered that certain microRNAs act as tumor suppressors, preventing uncontrolled cell growth, while others can actually promote tumor formation when dysregulated. And studies have shown that microRNA expression is often disrupted in cancer cells, either through the mutation of microRNA genes or through the changes in the pathway that control their production. On the bright side, researchers are now looking at microRNAs as potential biomarkers for early cancer diagnosis. Because microRNAs are so involved in regulating cell growth, changes in their expression can be an early indicator of cancer. MicroRNA is also being explored as a target for new therapies. By restoring normal microRNA function, we may be able to slow down, even stop the progression of cancer. And it's not just cancer. MicroRNA research is opening new possibilities for treating wide range of diseases, from cardiovascular conditions to neurological disorders. The last point is that while the therapeutic potential of microRNA is incredibly exciting, it is crucial to recognize that there are also risks involved. Just as with any powerful new technology, there are concerns that need to be carefully addressed before these therapies can be widely implemented. One of the biggest challenges with microRNA based therapies is ensuring that not only it is effective but also safe. MicroRNAs regulate a wide array of genes, and disrupting the balance in one system could unintentionally affect others. I think we must learn from our past experience. mRNA vaccines during the COVID-19 pandemic is a great example. We must understand that we are a product of billions of years of evolution, and we really don't have a clear idea how this body works. With microRNA therapies, we cannot afford to make similar mistakes. There is a real danger in rushing these treatments without fully understanding their long-term effects. That is why extensive clinical trials are essential to ensure that these therapies not only target diseases effectively, but do so without causing harm elsewhere in the body. In conclusion, the discovery of microRNA by Victor Ambros and Gary Ruvkun has revolutionized our understanding of gene regulation and development. These tiny molecules may be small, but their impact is immense. That's it for today. If you find this video interesting, consider subscribing to my channel and stay updated.